Okay, so we are here for Conversations with Faculty Part 2. And on the schedule today, we have Dr. Jesse Woodson and Rebecca Schomer from Plant Sciences, as well as Benjamin Renquist and Zelieann Craig from Animal and Comparative Biomedical Sciences. So we will go ahead and get started with Dr. Woodson. Go ahead and take it away.
Okay, thank you very much. So my name is Jesse Woodson. I'm a faculty member in the School of Plant Sciences. And the title of the presentation I'm giving today is called Understanding a Plant's Interaction with its Environment. And so in my lab, we're particularly interested in how plants sense their environment. And they have to be really good at it because unlike us, plants can't move. They have to spend their whole life rooted to where they had the bad luck to germinate. And so they have to be really good at being able to sense their surroundings and change to best fit their situation. And we are particularly interested in how chloroplasts and light is involved in this sensing. And what I'm showing you here, this is a transmission electron microscopy image of a chloroplast in a plant cell that's degrading itself when it becomes too stressed. And we think that these sorts of chloroplast turnover or degradation events are really important for plants to be able to acclimate and adjust to their environments to get rid of very damaged chloroplasts. And remember, chloroplasts are where photosynthesis occurs, where all the carbon fixation in plants occurs, and that they need to have healthy populations of chloroplasts. And so getting rid of the bad ones is going to be really important. So right here, I'm showing you maximum yields of several important crops that we grow across the world in this country, like corn and wheat and soybeans, sorghum, and how much we lose due to environmental stresses. And so the y-axis shows the maximum yield, but in orange and yellow is how much we lose due to abiotic and biotic losses. And you can see in particularly biotic losses, this is how much plant stress reduces our ability to grow crops. And what I want to point out here is that a lot of this comes down to what we call photooxidative stress, or stress that involves light, and that too much light in combination with drought or extreme temperatures leads to photooxidative damage, and that leads to a lot of crop loss. And so what we hope is that by understanding how plants sense these stresses and deal with photooxidative stress, perhaps we can actually increase our ability to increase crop yields and increase our ability to grow healthy, high quality crops. And so, as I said at my introduction, what we're really interested in is how plants sense their environment, and they have to be really good at it. Something that's going to grow in the crack in the sidewalk is still going to have to deal with all these different types of environmental stresses. And plants have many different ways to do this, and we've known this for probably thousands of years since we started agriculture. But as our techniques become more sophisticated, we're understanding that plants use organelles in their cells to do some of this signaling. A lot of eukaryotes do this. For instance, we use our mitochondria organelles to sense stress, but plants also use their chloroplast organelles. And they sense these sorts of stress or this kind of damage, and they send signals to the rest of the cell to regulate a lot of things, including growth, energy metabolism, programmed cell death. And in the case of mitochondrial signals in us, they can actually regulate aging and tumor progression. And so my lab is really interested in how organelles do this. And so I'm going to tell you about two major projects in my lab. And I'm looking to recruit undergraduate students to work on both of these projects. So the first one is understanding how chloroplasts sense these signals and initiate their own degradation. So here I'm showing transmission electron microscopy images of these degrading chloroplasts that we can actually see in plant cells, and they're being pushed into this structure right here. This is called the central vacuole. Here's a 3D image that we've made using 3D transmission electron microscopy techniques. And you can see the chloroplast in green right here, and these structures that come out of them in red that sort of pull out proteins from the chloroplast to be turned over. And so we're trying to understand how this works at a molecular level. And we take a molecular genetics approach. And so my group has tried to find mutants that block chloroplast degradation using this plant called Arabidopsis. And so we're very much a molecular biology lab or molecular genetics lab, trying to understand how this works. And so we know that when all these chloroplasts get degraded, that leads to cell death and you get a dead seedling. So we're finding mutants that block that. And we found lots of different mutations on the 5 chromosomes of Arabidopsis. And what we're doing right now is figuring out where those mutations are, what genes they're in, and then we can understand the proteins and genes involved in the signaling pathway. So this is I'm just showing here a table showing all the different genes that we're finding from these mutant screens, the kind of roles that they play, roles in chlorophyll synthesis, protein import into chloroplasts, chloroplast gene expression, ubiquitination, transcription. So it really allows us to study a lot of different aspects of cell biology and molecular biology and plant molecular biology. And so that is one major project we're working on. A second major project has to do with this collaborative group that just started a couple of years ago with the University of Illinois and Cornell University and the Boyce Thompson Institute, where we're trying to create what we're calling programmable plants that allow two-way communication between plants and people. And my group is leading efforts to do a synthetic biology approach to put genetic switches into plants so we can turn gene expression on and off using light. So just to back up a little bit, we're talking about biological switches. So what we are hoping to achieve as a center is this ability to communicate back and forth between plants. So plants are constantly stressed in their environment, but you can't always tell, or at least by the time that you can tell that a plant is overly stressed, it's too late. Like during drought stress, once it's wilted, it's probably too late to do anything about it, but it's stressed before you see any of that. So we're creating biosensors switches where a plant can turn on a switch when it's stressed, allow us, make something like a fluorescent protein or something that we can see, allowing a plant to speak to us or allow us to hear what a plant has to say. At the same time, we want to be able to talk back to the plant. And that's where my group is coming with these programmable switches to turn on gene expression that would increase plant health in a natural environment or on a field. So how we're going to do this? We're taking two separate approaches. One is we're taking natural photoreceptor proteins called phytochromes that can respond to specific wavelengths of light. And then we're engineering those to turn on specific genes within a plant. And so what's cool about these is that you can modulate them with different wavelengths. So you can turn them on with red light or turn them off with far red light. And so this is this non-invasive way to modulate gene expression within a plant without throwing on harmful chemicals, which is the old-fashioned way of doing these synthetic biology approaches. We're also making proteins that can make reactive oxygen species in the light, and those reactive oxygen species have signaling properties that we know can influence many things, everything from programmed cell death to modulating gene expression. And you might be wondering what this worm at the bottom is. We're also collaborating with engineers at Cornell that make things like soil swimming robots that can shine lights onto root systems, where maybe we can actually use these sorts of optogenetic switches, not only in above ground, but also below ground. So I'll end right there and just say that I think I have a very exciting group of people. We have undergraduates, graduate students, and professional scientists all working together on these two projects and a couple other smaller ones, but I'm mentioning the larger ones right here. And I'm absolutely looking to take an undergraduate or two over this summer. So thank you for your attention.
Thank you. Students or anyone else in the room, if you have questions, please feel free to ask. I'll take a minute and see if we have any.
Okay, well, thank you very much, Jennifer.
Okay, thank you. Thanks for being here. I really appreciate it.
Of course.
All right, so up next in our session today, we have Dr. Schomer, who's from Plant Sciences as well. And welcome.
Thank you. Yeah, sorry, I was a little late, so I'm glad I
No,
got here.
no problem at all. Hopefully you have powers to share. If you don't, let me know.
Let me see. Is that working?
Yes.
Are you guys seeing the slides or the?
We got it.
Okay, cool.
Okay, the floor is yours.
Right. Yeah. So hi, I'm Dr. Schomer, like I was just introduced, and I am going to tell you today about the work that we're doing in the School of Plant Sciences, where we're studying how bacteria are attracted to plant host roots. And some work that the goal that we're trying to accomplish is we're trying to understand the chemical language that is actually spoken between plants and bacteria in the soil. So some of you are probably wondering, what do I mean when I say bacteria are attracted to host roots? So to demonstrate that to you guys, I'm going to show you a quick time-lapse video that I made where of a common chemotaxis assay that we'd run in the Schomer lab. So what I'm showing you here in this square is a well of motobacterial cells that we've set up under the microscope between a slide and a cover slip and a U-tube. And this is at 40x magnification under dark field. And into this well of motobacterial cells, I've inserted a tiny capillary that contains about 3 microliters of agarose and a chemical that is potentially attractive to these motobacterial cells. And so this video has been collapsed into a few seconds from around 5 minutes originally. So as soon as we insert this capillary, what we see is the bacteria moving up this chemical gradient towards the tip of the capillary where this chemical is slowly being released into the well. And this is a phenomenon that we call chemotaxis. So a central question that my lab has about this process is, how are these bacteria perceiving these chemical signals? And then how are they then integrating these environmental signals into a behavioral or physiological response that allows them to locate and access a preferred host niche? So the model organisms that we use in the Schomer lab are phytobacteria, or bacteria that are associated with plants. And the preferred niche that we're looking at is host plant roots that are growing and actively dividing within the soil environment. And while they're doing that, they're exuding these really rich sources of chemical nutrients, nitrogen sources, carbon sources, all sorts of energy sources that these phytobacteria are sensing and swimming towards. Oh, shoot, I just realized that I don't have the right PowerPoint up, but I'll just continue. Let's see if hopefully, maybe I want to just skip right through this. Yeah, so I'm gonna go right here. Yeah, so one way that bacteria interact with their environment and basically interact with the chemical environment around their self in order to adapt and change their physiology and behavior depending on what their environment is telling them, is through these systems that we refer to as 2-component systems. And so, like the name suggests, 2-component systems have these 2 major components. First, we have a membrane sensor that is interacting directly with a chemical signal in the environment. And when that signal binds to our membrane sensor, the membrane becomes phosphorylated and it sends this conserved signal into the cell to the second component of the 2-component system, the response regulator. And this component actually interacts with a cellular target to change the physiology of the cell. So often that's turning on gene expression, turning off gene expression by binding to a promoter. And this phenomenon known as chemotaxis works in a really similar way. It's almost a 2-component system. It just happens to have a couple more components. So we still have the membrane sensor that is interacting with a plethora of different environmental chemicals. And it's sending this conserved phosphorylation cascade to our response regulator, key Y, which rather than interacting with promoters and changing gene expression is interacting with the flagellar motor protein. So the flagella is responsible for actually propelling this bacteria up or down a chemical gradient. And so based on the phosphorylation state of our response regulator, our flagella spins in a way that allows the bacteria to swim smoothly, or it spins in a way that causes the bacteria to twitch around and switch directions. So what makes chemotaxis really interesting and complicated is that, unlike your traditional 2-component system where we have one membrane sensor that interacts directly with stringently with one response regulator. What we have in chemotaxis are multiple chemoreceptors, so multiple membrane sensors that are interacting with a lot of different environmental signals, and they're all feeding into this same response regulator and the same output. So we have multiple inputs for one output and behavioral response, which makes it really complicated and difficult to understand the hierarchy of how these bacteria are following different complex chemical signals like those from host roots and complex environments. So this example that I'm showing you here with the four receptors is actually a relatively simple chemotaxis system as well. So this example is actually from an animal pathogen like E. coli, and because of the host physiology of animal pathogens, they don't really rely on their chemotaxis much to get through their life cycles. So they tend to have a low number of chemoreceptors, around 4 per genome. But if we look at phytobacteria, which are the organisms that my lab is really excited and interested in studying, like Ralstonia solanacearum, this is a plant pathogen that's causative of bacterial wilt disease. This is where my slides got messed up here. I'll just go back and show. So it's causative of a disease called bacterial wilt disease, and which I'm showing you here, which causes millions of dollars of losses for farmers across the world who farm things like solanaceous vegetables, bananas, a variety of different botanical host families. So on the left here, I'm showing you a plant that's been watered with water that's infested with Ralstonia solanacearum. And on the right, you can see this healthy plant that's full of turgor and very happy. So this plant pathogen infects the plant through the soil. It lives first in this free living environment where it's basically scrounging around for any chemicals that it can find, using its chemotaxis to locate little pockets of nutrient-rich areas, and ultimately following chemical gradients to colonize the host plant. And once it accesses a host root, or locates and discovers a host root, it enters into the plant and is spread systematically throughout the plant using xylem flow. And so it no longer needs its flagella there because it's using the host physiology to move throughout the plant. And it generates these thick biofilms that ultimately end up wilting the plant, causing us to lose causing fruiting loss, causing ultimately death of the plant. And then the Ralstonia escapes again into the soil, where it starts to live in this metabolically versatile state again, seeking out these ideal carbon and nutrient sources. Sorry, let me skip back to where I wanted to be. So because of that, because of their need to find many different carbon, nitrogen, energy sources, Ralstonia has this metabolically versatile physiology. And we then see that it has a high number of different chemoreceptors that are able to interact with a much wider range of environmental chemicals. And this is really common of organisms that are free living in the soil, free living, or that are associated with plants that need to go through this search for their host, for the ideal environment, rather than those that are host restricted. So one last very interesting thing about Ralstonia, and the main drive of my research program, is I'm trying to understand how this chemoreceptor repertoire, the ability to sense so many different chemical compounds, might influence how these organisms actually discover and locate the appropriate hosts in the soil. So here I'm showing you a phylogenetic tree of the Ralstonia species complex, which actually contains thousands of different strains that we've isolated from dozens and dozens of different botanical host families. And so across the Ralstonia species complex, there's a really wide host range. So these organisms are able to infect a wide range of different botanical hosts. But if we start looking at individual strains, what we find is that the host ranges are unique and can be more strict. So for example, highlighted in blue, I have this strain IBSBF 1503. That's a strain of Ralstonia that infects a really wide range of different host families. So you'll see that it infects, solanaceous vegetables, it infects bananas, it infects Brassicaceae, and it also infects, ornamentals and a really wide range of different families that this organism has been isolated from. Whereas GMI 1000, you can see that's highlighted in purple, only affects a handful of these botanical host families. And then we have a strain highlighted in green, PSI 07, that only infects a couple different families. So it's only able to infect solanaceous vegetables and bananas. And so this variation in host range, our hypothesis, the hypothesis of the Schomer lab, is that this variation in host range is dictated by the chemoreceptor repertoires of these different strains and the different chemicals that they're able to detect and follow in the soil to locate host roots. So to summarize, the goals of the Schomer lab are to understand the rules of chemotaxis across these different scales of biology. So all the way from the ecosystem level, where we're trying to understand what are the chemicals that are leading these plant pathogens and commensals to different host plants, all the way down to looking at specific receptors in these organisms and linking them to specific chemicals as well. So we're looking at the ligand receptor specificity as well. So if you join my lab this summer, we'll be working on a lot of projects in this trying to understand this plant pathogen and how it interacts with specifically tomato. And you'll develop molecular biology techniques to try and generate chemoreceptor mutants in this Ralstonia solanacearum strain, Ralstonia solanacearum organism, which I'm showing here on the right. It's this very beautiful pink color. It's very goopy when you grow it on a plate. And we'll also be studying the bacterial behavior of Ralstonia using microbiological assays like that one I showed you at the beginning of the presentation to probe the different chemical attractions and preferences of this organism. And also you'll get a chance to be part of a brand new lab I just started earlier this year, and you'll get to learn a little bit about how a lab is built and put together. So with that, I will open it up for any questions that people might have. My contact information is on here, as well as my Twitter or my email. So looking forward to hearing what you guys have to say.
All right, students, if you have any questions, feel free to fire away. And as Dr. Schomer mentioned, of course, you can reach out to her directly, separately if you would like. So
sorry about that.
No problem at all.
Slides.
I'll give the students like
right.
10 seconds.
Okay.
Very quiet today. It must be the snow.
Yeah, I totally get it. All right. Thank you so much,
thank
everyone.
you. Yeah. Thanks so much for being here. Up next, we have Dr. Renquist from Animal and Comparative Biomedical Sciences. And I believe I have given you sharing power. Let's see if that works for you. Oh no, I see you speaking, but I do not hear you speaking. We do see your slides though.
Okay, I'm going to be speaking through my phone because my computer, when I use it for audio, just is absolutely terrible.
Okay, perfect.
So, and I think I'm sharing the PowerPoint.
Yes, we can see it well. Great. Floor is yours.
So my lab's focused on obesity and obesity-associated diseases, specifically diabetes and hypertension. And diabetes and obesity affect about 30% of the population, but they affect even more than that. In fact, in Arizona, 1 out of every 2 people is insulin resistant, meaning they haven't yet developed type 2 diabetes, but they're pre-diabetic. And that can cause great health concerns. Amputations arise from that. We have additional issues like poor vision, health concerns, cardiovascular disease. And in fact, the same thing happens with hypertension. About 30% of the US population is hypertensive. And we can see that we will notice that there's a pattern down here along the southeast portion of the United States, where there's higher incidences of obesity, and then there's higher incidences of type 2 diabetes and hypertension. And so the questions we ask is, how do the organs communicate with each other to maintain homeostasis in a normal animal, or induce pathophysiologies, meaning bad physiologies, in an obese setting? And can we manipulate that organ-to-organ communication to develop effective therapies for type 2 diabetes and for hypertension. And I just want you guys to jump in at any point if you have questions. I don't mind being interrupted. So one of our fun projects that we're working on right now is in collaboration with an investigator at Ohio State University who developed a way to make the heart beat better. And you guys have all gone and run and your heart starts beating more, but your blood pressure doesn't really change that much. And how is that possible? Well, the way that occurs is in the aortic arch, which is right here, there are nerve endings that sense how much pressure the blood is making coming out of the heart. And if there's a lot of pressure coming out of the heart, you vasodilate all the blood vessels in your skeletal muscle to alleviate the change in blood pressure that would be caused by going for a run or playing soccer or watching a football game that's driving you crazy. Okay, so we decided to take advantage of that, and there are therapies currently that work to affect how the heart beats. Obviously, those could be used. We started with a model where we induced expression of a very special troponin C molecule, and that's one of the contractile proteins involved in heartbeat. And by making a mouse express a troponin C that allows the heart to beat better, we were able to take obese mice and make them more insulin sensitive. This test is a test where we inject the mouse with insulin and we see how much blood glucose goes down. And in those mice that were expressing this troponin C that made the heart beat better, we had improved insulin sensitivity. And that's because what we did is we shifted blood flow. So this is a measure of blood flow, and we set our controls to 100%, and we look at everything relative to that. You can see we improved blood flow to gastrocnemius muscle, to soleus muscle, to the stomach, to the epididymal white adipose tissue. And so just by making the heart beat better, we improved blood flow throughout the body. We caused vasodilation and we improved insulin sensitivity. And this isn't what we would typically think of as a treatment for type 2 diabetes. People would typically start to think about treatments for type 2 diabetes where you're trying to make the cell that responds to insulin more insulin sensitive. It turns out insulin resistance has very little to do with the cell being responsive to insulin. It has more to do with getting the insulin to the correct place. And that's just like any job. You know, you can be great at your job, but if you don't show up to your job, you aren't doing a good job at it. Well, that's the same with insulin. If we can't get the insulin to the cell that matters, then we can't allow it to be effective. Another organ communication that we're looking at is how does the body know that fatty liver occurs? So if you and I don't eat for a while, and I'm talking like 72 hours, we will have fat buildup in our liver and we will become insulin resistant. And that's exactly what we want to have happen, because when we haven't eaten for 72 hours, we don't want insulin to be having action. However, in obesity, now we have fatty liver all the time, and this leads to insulin resistance. But the question we were asking is, how does skeletal muscle, which is the place that takes up glucose, how does it know that the liver is fatty? So how is the liver communicating that? And so we know it is communicating it based on studies from the 70s and 80s. It's important to realize in science that we're building upon knowledge that was created 20 or 30 years ago, not our own knowledge. And that everything, actually, I teach biochemistry, everything you guys are learning in your classes is stuff that 50, 60 years ago, people didn't even know about. I mean, if we think about, you know, the discovery of DNA, it wasn't that long ago that we discovered how DNA. Okay, so if we cut the nerve that runs from the liver to the brain, insulin goes up. That's what this figure is showing here. If we stimulate this nerve, insulin goes down. That tells us this nerve that runs from the liver to the brain is really important. You'll remember that same nerve was in the aortic arch. So we're really focused on peripheral nervous system control of metabolism. If you cut the nerve that runs from the liver to the brain, you also affect skeletal muscle blood flow. And you affect that by changing the peripheral nervous system. So you'll notice that there are some common themes. Blood flow is a big one. And in fact, if you cut the nerve that runs from the liver to the brain, you cause insulin resistance. Skeletal muscle is specifically, the uptake of glucose by skeletal muscle is much, much lower. So we wanted to understand how fatty liver could be changing all of these pathways. But the first thing we had to figure out is, what is fatty liver doing to change the activity of this nerve? I'm going to skip that. And for those of you who like biochemistry, this is the TCA cycle. And in the TCA cycle, succinate goes to fumarate, goes to malate in the liver. But what happens in obesity is succinate can no longer go to fumarate. So succinate actually has to go to succinate semialdehyde, which then produces the inhibitory neurotransmitter GABA. And our lab showed that in obesity, the amount of GABA that the liver releases increases. And GABA inhibits the activity of that nerve that runs from the liver to the brain. And so we are really focused on understanding how that works. I've had undergraduates involved in these projects throughout. In fact, we've got a project right now where we're developing inhibitors of GABA transaminase. And what's neat about that project is it was started by 2 undergrads, one in my lab and one in a lab in chemistry. We just got a $2.5 million grant to fund that research. The student who did it from my lab is now a PhD student in chemistry at Stanford. And the other one is a PhD student at Harvard in chemistry as well. So we've had some real success in taking undergraduates, giving them projects that are much, much more complex than the undergrads might have thought at the beginning that they could handle. But they've yielded amazing results. They've helped us gain funding, and we've helped those students get to where they want to go as well. I don't want to take any more time. I'm happy to answer questions. There's a lot more here, but I feel like I don't want to overwhelm you with more information than you need. The real focus of our lab is how obesity changes metabolism to cause hypertension and diabetes.
Perfect. Thank you. Students, if you have any questions, we'll give you like 10 seconds. You can pipe up and ask what you're thinking.
I'm going to pull up the front slide so you have my email again.
Thank you.
Wonderful.
Well, thank you so much.
Thank you, Jennifer. I really appreciate it.
No problem. Take care.
Thanks.
All right. And
good luck, Dr. Craig.
Thank you. Great to see your talk.
Thanks.
And certainly last but not least, we have Dr. Craig, who's also here from Animal and Comparative Biomedical Sciences. So thanks for joining us.
All right. Thank you. Thank you for the invitation. Should I, let's see, get permission for sharing?
I think hopefully you have it, but maybe not. Let's see.
Maybe I do now.
Try now.
Now it's working.
Okay.
All right. So I will just need confirmation that once I start the slideshow that I'm sharing the right one. You seeing the slides? Okay, I see thumbs up. Well, hi. So are these the students, Olivia and Dasha? Is that correct?
Yes. And we're recording as well. There's a few students who wanted to be here but have a class. So if you're okay with that, then we'll record and share that way.
Yes, great. Well, this is always a very exciting time for me to come and share with all the, the undergraduate programs, my research. I actually did undergraduate research here at U of A. Oh my gosh, 20 years ago. So that put me in a really interesting trajectory that led to what I'm doing now. So I'm associate professor in the School of Animal and Comparative Biomedical Sciences, but I'm also assistant dean for research for the College of Agriculture and Life Sciences. So that one experience really sent me into a trajectory that has led me to very interesting places. So I titled my talk, "It's Not You, It's the Environment." It's kind of like a joke back to when I realized that I really was interested in reproduction and how the environment can shape our ability to reproduce. And what really keeps me coming back to work every single day, despite any adversity or stressors, is that I really dream of a future where there are healthy women that can have healthy babies. I am a mother myself, and I was a baby too, so I'm glad somebody had me. And contrary to what most people like to represent, kind of like us at the beginning, right before fertilization, they tend to represent humans as a sperm, like the one that made it, this faster that swam. Actually, most of what we become comes from the egg. And that comes from mom. So you're not the fastest to swim, you're the one that got out of the ovary and was fertilized. So that's kind of represents more of what we are. So I'm a toxicologist by training. So how do I contribute to healthy women and healthy babies? So I focus on chemicals in the environment that can actually interact with the reproductive system. And the specific type of chemical that I have been working on are called phthalates. You might have heard of them. They have hit the news quite a few times right after BPA, for example, bisphenol A, which was another really famous one. And so these phthalates are chemicals that have a very specific structure that allows them to be fat soluble. So they're used in a lot of things. And when they get into the body, they actually go everywhere. They are used in lots of different products. They are present in, they exist in many different chemical structures. So that's what makes them be so ubiquitous, is because they have all these different structures that can be used in things like plastics, but they can also be in makeup and the coating of some medications. So we know they're everywhere. So we're definitely interested in them. Actually, human studies have shown that phthalates can be associated with adverse reproductive health outcomes. So basically fertility issues. And I've shown some examples here. For example, when women go to the clinic and they have work, they're setting themselves up for assisted reproduction. We actually get access to a lot of fluids from the body. So we get access to urine, fluid from the ovaries and the eggs. And so we learn a lot about what their exposures can be. And so we know that women that have higher level of phthalate markers in their urine have an increased risk for early menopause, reduction in their dynamic ovarian reserve. So these are in the ovary. We have these units that contain the eggs. They're called follicles. So the higher the level of phthalates in their urine, the lower the number of eggs these women have available for reproduction. And when they are subjected to hormone treatments at the clinic to ovulate, they don't ovulate as well. And finally, when they become pregnant, if they have a high level of phthalate markers in their urine, a lot of them lose their pregnancy early, so they don't have a successful pregnancy. So as you can imagine, this is very, obviously very depressing news, and really bring phthalates and other chemicals that can do this really up to the top of a priority list, because remember, we want healthy women and healthy babies. The phthalate that I'm going to show you some examples of the research that we know is called dibutyl phthalate. I call it DBP for short. And dibutyl phthalate, actually, we have a really good handle on how much the general population is exposed to this chemical in different settings, like the general population, occupational exposures, medications. We know that these, the levels of markers of this phthalate exposure are higher in women. And unfortunately, the original studies that characterize these chemicals did not use doses that were low enough to mimic what humans are exposed to. So that's why we missed all the issues that these chemicals can bring about. So sometimes students have that question and say, well, if this is so bad, how did it even get into products? It's because originally we test really high doses that don't represent what humans do. And when we have compounds that can mimic hormones, like phthalates and bisphenol A, for example, they actually can trick our toxicity experiments and make us think that they're safe when they're actually not safe when you go really, really low in the range where hormones actually work. Okay, so using all this information, I pretty much launched my lab 10 years ago with two main driving questions. And the first one was, can these exposures and the associations, the health associations that come with them, be replicated in a mouse model, right? We can't do experiments with humans, but we can find out things with animals in the lab. So I wanted to know, can I replicate this in a mouse model so I can study it? And the second question was, are these associations mediated via direct effects on the ovary? I'm a toxicologist. The ovary is my organ. That's the organ that I'm really excited about to study. So I wanted to know, are there any effects that they're seeing in people mediated via the ovary? So I launched the Craig Lab. We specialize in reproductive toxicology. So I wanted to show you 3 different approaches that we have used over the years to, pretty much enhance our understanding and model these things in animals. So we have been modeling human relevant chemical exposures and the effects associations in mice so that we can get insights into the mechanisms in humans. So the first approach that we've used is looking at ovarian follicles. These are the little cell compartments that have the egg, and they are in the ovary. These produce estrogen, and they actually release the egg so that there can be a fertilization. So we actually take those and expose them in vitro, and I'll show you an example of what data looks like from that. The second approach is toxicokinetic studies. So toxicokinetics, what tells you toxicant, kinetic movement, how toxicants move through an organism. How are they absorbed? What organs do they go to? What does your body do to them? Transform them into other metabolites, and how they're excreted. For us to be able to model these chemical exposures in mice, we need to understand how they move through the mouse and what are the differences between that and humans, and how much I need to give a mouse so that it looks like a woman, in terms of the toxicity that is produced. And the last approach that I'll share with you today is what we call preconceptional in vivo exposure. So we use whole animals and we give them doses that are similar to what women can experience. And then we use them for our studies to look at all those things that I said at the beginning, the amount of follicles that they have available for reproduction, the quality of their embryos, are they able to ovulate in a way to replicate what humans do. And once we achieve that, we actually look at mechanisms and figure out how that works. And one thing I really wanted to add, I have it at the end too, the names of the students, but this research has been supported significantly by undergraduate students in the EHS True program. And actually, one of them is already a co-author in one of our publications, and we have another one that is a co-author in a current publication right now. By the time that we're done with those experiments, I think our most recent student, Miley, might be in more than one publication in the end. So very excited. And they always bring a lot of fabulous energy to the lab. So here are the examples I was going to show. So I talked about direct exposures in vitro, and this is an example that we did with dibutyl phthalate, an experiment where we hypothesized that if we give ovarian follicles that we have on a dish a treatment with this phthalate, that their development will be impacted, it will be inhibited. So these are 3 examples of data that we get from such a study. The first one is in vitro growth right here. We see increasing concentrations of the phthalate on the x-axis, and on the y-axis, the percentage of follicles that were still growing after 3 days in culture. And the first thing we see is that, at the lower levels, the follicles are doing similar to the control, which is our yellow line. But if we continue to increase that concentration of phthalate, the follicles start to not do very well. And if we take them out and we process them and image them with a specific stain for cell death, we can see that they actually are accumulating dying cells. So these follicles are dying. So these concentrations actually are lethal to ovarian follicles. And this graph here shows you that we can actually look at things visually with imaging, but we can also quantify them and generate graphs that allow us to do statistical analysis on them and make some conclusions. And this work was published, and we had actually a student help us with us, an undergraduate student, not from EHS True, but also from another U of A program. The second example here is an example of a toxicokinetic study where we wanted to check, right? For example, if we treat animals with DVP, which is the phthalate that I've been talking to you about, will we detect metabolites or part of the breakdown of that compound in the ovary of mice. So we use whole mice, we gave them DVP, but then we were checking for another compound in their ovary. And so here's an example of the data. Before I show you that, just to kind of have a little more visual explanation of it. So dibutyl phthalate, once an animal ingested or a person ingested, the cells in the gastrointestinal tract will actually clip one of these two chains and produce this metabolite. It's called monobutyl phthalate. So that will circulate through the body and we can detect it. So we did a couple different experiments. In the first one, we gave them one dose of 3 different levels of DVP or oil, and then we track the levels of MBP in the ovaries of the mice, right? And you'll see initially for the really high dose, this is a really high dose, we gave them a whole bunch of it, and the concentration went up really high, and then over time started to go down. So that gives you an idea of how long does this compound linger in the ovary, right? So that we know how much gets there, so that we can match that to the human studies. We also saw that when we repeated dosing, so we give 10 repeated doses daily, actually the levels actually drop much faster than when we give only one dose. So this shows adaptation, that the liver and the different tissues in the body actually adapt to receiving that chemical exposure, and they actually clear it faster, right? There's the joke that say the more alcohol you drink, the faster you get metabolize it, or you can withstand it a little more. I don't endorse that, but that's an example that most people can relate to. And the last one that I wanted to show you, it's on this branch of my work, doing preconceptional in vivo exposure. So here we expose a whole animal to the phthalate, and then we look at the phenotypes that are known to happen in women from the epidemiological studies that I showed at the beginning. I've actually selected to show you some of our work looking at embryos. So in this experiment, the animals were treated for 9 days, and then they were stimulated to ovulate with hormones. They were put with a male to get them fertilized. And then the animals were euthanized and the embryos collected. And over the next 3 days, we were looking at the development of those embryos. And so we divided them into 3 different groups: embryos that basically did not go forward after fertilization, embryos that actually became fragmented, so something in their development went wrong and they were able to continue organized division, or embryos that looked either like a morula, which is this little ball of lots and lots of cells, or better development, which are blastocysts. These fellas here are actually ready for implantation. So when we go to the clinic and you undergo an in vitro fertilization, they will generate blastocysts, and these are the ones that will be put back into the uterus and to see if we can get a pregnancy. So when we divided our embryos into these 3 groups, we found that the 2 groups of animals that were treated with 2 levels of dibutyl phthalate that are similar to those that occur in women actually were increasingly fragmented. They were a great proportion of them were fragmented. So this definitely gives us a clue that we might be able to model the embryo loss that occurs in humans with this animal model. And so I didn't include a lot of the other experiments that we do with mechanisms. I just kind of wanted to give a little flavor and example of each one of the different areas that we are working on. So I'm always acknowledging my lab. We are at the Bio5 buildings in BSRL, which are part of the Bio5 campus. And these are some pictures of my students, sources of funding, and the names of past EHS True trainee contributors that we've had. So over the past 10 years, we actually had the pleasure of working with 4 students from that program. And I'll take any questions.
Thank you. So we'll wait a few seconds if there's any questions. And of course, students can always reach out to you individually if they would like.
Yep.
Well, Dr. Craig, thank you so much for your time. I really appreciate it and for your presentation, and we will send students your way.
Thank you. Thanks for the invitation. Have a wonderful rest of your week.
You too. Take care.
Yeah,